What causes blood coagulation? See, coagulation means clotting. So clotting in our body happens because of platelets. Okay, platelets. Now, you know that when you cut your finger or hurt yourself, your wound does not continue to bleed for a long time. Usually the blood stops flowing after some time. Do you know why? Because blood exhibits coagulation or clotting in response to any injury or trauma. This is a mechanism to prevent excessive loss of blood from the body. You have observed a dark reddish-brown scum formed at the site of a cut or an injury over a period of time. It is a clot or a coagulum formed mainly of a network of threads called fibrins. So the blood clotting which happens in a body, it happens because of the presence of fibrins. Fibrins are proteinaceous stuff. Now, it is a clot and coagulum formed mainly of a network of threads called fibrins in which dead and damaged formed elements of blood are trapped. Fibrins are formed by the conversion of inactive fibrinogen in the plasma by the enzyme thrombin. There is an enzyme thrombin which activates the fibrins, or it activates the fibrinogens and forms the fibrins. Do you remember fibrinogens, albumins, and globulins? These three things were present in the blood.
Yes, ma'am.
So they get activated and get converted into fibrins by the enzyme that is known as thrombin. Now, thrombin in turn are formed by another inactive substance present in the plasma called as prothrombin. Then this thrombin is not present as an active enzyme. It is actually present initially like an inactive one that is known as prothrombin. Now, this also has to get activated in order to activate fibrinogen. So, an enzyme complex which is known as thrombokinase is required for the above reaction. So, for the conversion of prothrombin into thrombin, there is an enzyme that is thrombokinase which is required. Now, this complex is formed by a series of linked enzymatic reactions or a cascade process. Cascade process is just like a waterfall, one after the other, the processes keep on happening, which involves a number of factors present in the plasma in an inactive state. An injury or a trauma stimulates the platelets in the blood to release certain factors which activate the mechanism of coagulation. So, as soon as the person has any injury, a cut or a wound, then what happens? The platelets get activated, and what they do is that they release certain factors to start the process of clotting or to start the process of coagulation. Now, certain factors released by the tissues at the site of injury can also initiate coagulation. Calcium ions play a very important role in clotting. Calcium dipositive ions, they are also very important for clotting mechanism. So, have you understood clotting?
Yes.
You understand how as soon as we get a wound or as soon as we get an injury, first of all, our platelets get activated so as to start the clotting mechanism. At the same time, the tissues also start releasing stuff to initiate coagulation. So double responses are collected, and then the whole process of fibrinogen like the inactive fibrinogen getting activated by what? By thrombin and getting converted into fibrin. Then same way, first of all, the thrombin has to get activated. So it gets activated from prothrombin by the enzyme that is known as thrombokinase. So the whole process of coagulation clear to you?
Yes, ma'am.
Any doubts?
No.
Okay. Now we know that our fluid connective tissue is blood. But apart from that, apart from the blood, there is also lymph which is present in our body, and this lymph is a colorless fluid and it is also known as tissue fluid. As the blood passes through the capillaries in tissues, some water along with many small water-soluble substances move out into spaces between the cells of tissues, leaving larger proteins and most of the formed elements in the blood vessels. This fluid released out is known as interstitial fluid or tissue fluid. This is the lymph. Imagine it like a fluid that has been filtered out. Okay, that is tissue fluid. Now, it has the same mineral distribution as that in plasma. Exchange of nutrients, gases, etc. between the blood and the cells always occur through this fluid. So the sole purpose of the lymph is to actually exchange nutrients and exchange gases in between the cell and the blood. An elaborate network of vessels, which is known as the lymphatic system, collects this fluid and drains it back to the major veins. The fluid present in the lymphatic system is known as the lymph. Lymph is a colorless fluid containing specialized lymphocytes, which are responsible for the immune responses of our body. Lymphocytes, there are B lymphocytes, there are T lymphocytes in our body. So these lymphocytes are actually present in the lymph and they are responsible for the immune responses of our body. Lymph is also an important carrier for nutrients, hormones, etc. Fats are absorbed through lymph in the lacteals present in the intestinal villi. So if we see in totality, exchange of nutrients, exchange of gases, then carrier for nutrients, carrier for hormones, and then also for the absorption of fats in the intestine. These are the functions of lymph. Okay, the functions of lymph can be asked in the exam. Is it clear? Lymph is clear?
Yes, ma'am.
Oh. All right, now moving on to the circulatory pathway. Now see, we have like studied in animal kingdom that the pathway or the circulation can be of two types. It can be open type of circulation and it can be closed type of circulation. Now, when it is open, it means that the organs are bathed in the blood, right? There is no connecting or no, you can say, vessels present in the body of the organism. So for those organisms that have this kind of circulatory system, they are known as like what? They are the lower organisms and they contain the open circulatory system. After that, there is the closed circulatory system in which the blood is pumped by the heart and it is transported to the entire body with the help of certain vessels such as arteries and veins and capillaries. Now, the circulatory patterns are of two types, open and closed. Open circulatory system is present in arthropods and mollusks, in which blood pumped by the heart passes through large vessels into open spaces or body cavities known as sinuses. Here, all the organs are bathed in the blood. Annelids and chordates have a closed circulatory system in which the blood pumped by the heart is always circulated through a closed network of blood vessels. This pattern is considered to be more advantageous as the flow of fluid can be more precisely regulated. Once it is traveling through the blood vessels, so the fluid can be better regulated in that way. That is why closed circulatory system is much more advanced or better than the open circulatory system. This part is clear?
Yes, ma'am.
Okay, all vertebrates possess a muscular chambered heart. Our heart is made up of muscles, more importantly, the cardiac muscle. Fishes have a two-chambered heart with an atrium and a ventricle. Amphibians and the reptiles, I mean all the reptiles except for crocodile, they have a three-chambered heart with two atria and single ventricle. Whereas crocodiles, birds, and mammals, they possess a four-chambered heart with two atria and two ventricles. In fishes, the heart pumps out deoxygenated blood, which is oxygenated by the gills. So once the heart is pumping, it will pump out deoxygenated blood, and that deoxygenated blood will reach the gills so that it can get oxygenated. And then it is supplied to body parts from where the deoxygenated blood is returned to the heart. So the type of circulation in the fishes is known as single circulation. Now talking about in amphibians and reptiles, the left atrium receives the oxygenated blood from the gills or the lungs or the skin, wherever oxygenation happens. So from there, the left atrium or the left auricle receives the oxygenated blood, and then the right atrium gets the deoxygenated blood from the other body parts. However, since there is only one ventricle, it gets mixed up. For example, this is one chamber, this is another chamber, and this is the one ventricle. So if this is left and this is right, so the oxygenated blood is coming here, the deoxygenated blood is coming here, and after that it is getting mixed because there is only one ventricle. Now, in the case of the amphibians and reptiles, mixed blood is pumped out, mixed blood. So this type of circulation is known as incomplete double circulation. Double circulation, why? Because in the two upper chambers, blood from different areas are getting collected, but it is getting mixed. That is why it is incomplete. In birds and mammals and crocodiles, oxygenated and deoxygenated blood received by the left and the right atria respectively pass on to the ventricles of the same side. So now in us, 4 chambers are there. This is the left atria. This is the left ventricle. This is the right atria and this is the right ventricle. The oxygenated blood will come here. The deoxygenated blood will come here. From here, it will go here. And then it will have a separate places where the right ventricle and the left ventricle pump these blood out. So here, there is no mixing of blood. The oxygenated blood and the deoxygenated blood, both of them are separated. So we'll call this type of circulation as double circulation. Complete double circulation. Is this clear?
Yes, ma'am.
Any doubts in single circulation, incomplete double and double circulation?
No, ma'am.
Okay. Now talking about the human circulatory system. Human circulatory system, it is also known as blood vascular system. It consists of a muscular chambered heart, a network of closed branching blood vessels and blood, the fluid which is circulating. Now, heart is a mesodermally derived organ. You remember mesoderm, ectoderm, endoderm?
Yes, ma'am. The innermost layer, mesoderm.
Mesoderm was the middle layer.
Middle layer. Yes.
So, middle and what layer? Which layer was it?
Sorry.
Which kind of layer are the ectoderm, endoderm, and mesoderm? Like what kind are they? Hmm? Yes, what kind of layers are they?
Germ layer.
They are germ layers. Correct. So the germ layers, out of the 3 germ layers, the middle one, that is the mesoderm, this is from where the heart is formed. Mesoderm gives rise to the heart. Now moving forward. Now the heart is situated in the thoracic cavity in between the 2 lungs, and it is slightly tilted to the left. It has the size of a clenched fist. It is protected by a double-walled membranous bag, which is known as pericardium. The layer surrounding the heart is the pericardium, and it encloses the pericardial fluid. Our heart has 4 chambers. two relatively small upper chambers which are known as atria or auricles, and two lower chambers that are larger, they are the ventricles. Now, see. Okay, they are saying that the heart is tilted to the left. Do you know why? Why is the heart not straight present in between the lungs? Why is it tilted towards the left side a little bit? Why do you think so? There must be a reason, right?
Uh, taking sufficient space. Can you hear me now? There's no sufficient space
Okay. Why?
because of the presence of rib cage.
Because of the
Presence of rib cage.
not actually rib cage, because of see, it is slightly tilted towards the left side because the right lung is a little larger in size as compared to the left lung. Okay, that is it. Okay. The
Yeah.
right lung, like the lungs are right and left. So the right one is a little larger, in size. Now, this is I have drawn the chambers and boxes for the ease of understanding. Now, a thin muscular wall, which is known as the interatrial septum, separates the right and the left atria. The interatrium, the inter means in between. So the wall that will be present in between the two atria will be known as the interatrial septum. Whereas a thick walled interventricular septum separates the left and the right ventricles. The atrium and the ventricles on the same side, meaning here, they are also separated, and they are separated by a thick fibrous tissue which is known as atrioventricular septum. So imagine septum being like a wall and it is separating each of the chambers. However, each of these septa are provided with an opening through which the two chambers of the same side are connected. Obviously, since the blood has to pass on from one chamber to another chamber, so it has to be connected in some way, and that connection is happening because of some opening is there. Now, the opening between the right atrium and the right ventricle, it is guarded by a wall formed of three muscular flaps or cusps, and it is known as tricuspid wall, meaning there are three walls that are sort of, for example, if it opens, blood flows down, and if it closes, blood cannot flow back up. So that is why walls are there. So the walls between the right auricle and the right ventricle is the tricuspid wall because it is made up of three cusps. Okay, now there is a bicuspid, or the other name for bicuspid is mitral wall. It is made up of two cusps and it is present at the opening between the left auricle and the left ventricle. Okay, now the openings of the right and the left ventricles into the pulmonary artery and aorta respectively are provided with the semilunar walls. Again, certain walls are present so that when the ventricles pump out the blood, they will get transported into the pulmonary artery and aorta, and they are also guarded by a wall. The walls in the heart allows the flow of blood only in one direction, that is from the atria to the ventricles, then from the ventricles to the pulmonary artery or aorta, meaning the blood flow has to be in one direction. It cannot go back. Okay, if it goes back, then it is a very serious condition. That is why walls are present. Walls prevent the backflow of the blood. Okay, it like stops it. It like whenever backflow will happen, it will lead to a very serious illness or anything. So that is why backflow is prevented with the help of the walls. Clear?
Yes, ma'am.
Now the entire heart is made up of cardiac muscles. The walls of the ventricles are much thicker than that of the atria. A specialized cardiac musculature, which is known as the nodal tissue, is also distributed in the heart. A patch of this tissue is present in the right upper corner of the right atrium, which is known as sinoatrial node, SAN. Upper right of the right atrium. Another mass of this tissue is seen in the lower left of the right atrium, close to the atrioventricular septum, that is known as atrioventricular node. There are two types of nodes that are present, sinoatrial node and atrioventricular node. Now, a bundle of nodal fibers, atrioventricular bundle, that is the AV bundle, meaning the veins or the arisings from the nodes, they are the bundles, continue from the AVN, which passes through the atrioventricular septa to emerge on top of the interventricular septum, meaning, you see this, it is emerging on top of the interventricular septum, the yellow part that you are seeing. Okay, now, and then it divides to right and left bundle. It is sort of branching out to right and the left side. These branches give rise to minute fibers throughout the ventricular musculature, meaning throughout the muscles of the ventricle on both the sides, and they are known as the Purkinje fibers. The left and the right division and the minute division throughout the whole entire ventricle is known as the Purkinje fibers. These fibers, along with the right and the left bundles, are known as bundle of His. You see here in this particular diagram. See, it is first the AVN is present here, and then the bundles that are arising from it is actually first transported onto the interventricular septum, and then it is separating and sort of like branching out to the whole muscle of the ventricle. And this Purkinje fibers, along with the right and the left bundles, they are known as the bundle of His. Now, the nodal musculature has the ability to generate action potentials without any external stimuli, meaning it is auto-excitable. See, our heart can pump on its own. It does not need anyone to tell it to do so. Why? Because of these nodes only. These nodes help to generate action potential. Action potentials are just like regular beating of the heart. They happen because of that. However, the number of potentials could be generated in a minute. They can vary different parts of the nodal system. The sinoatrial node can generate the maximum number of action potentials, that is 70 to 75 per minute, and it is responsible for initiating and maintaining the rhythmic contractile activity of the heart. Therefore, it is known as the pacemaker of a heart. Sinoatrial node, meaning any beating activity in a heart, the initiation right from when life starts. The SA node tells the heart to beat, because of which the heart keeps on beating. So it is the pacemaker. Okay. And our heart normally beats 70 to 75 times in a minute. The average beating or the average heartbeat is 72 beats per minute. In 1 minute, 72 times the heart would contract, relax, contract, relax. So this is your human heart for the whole entire circulatory system and how it happens. Do you have any doubts?
No, ma'am.
This whole thing is clear?
Yes.
Okay, now talking about the cardiac cycle. How does the heart function? Let us take a look. To begin with, all the 4 chambers of the heart are in relaxed state, meaning they are ready for the blood to fill. Now this relaxed state is known as joint. Why joint? Because all 4 of them are together in this relaxed state. They are in joint diastole. Diastole means relaxation. Systole means contraction. So they are in joint diastole. Now, as the tricuspid and the bicuspid walls are open, blood from the pulmonary vein and vena cava flows into the left and the right ventricle respectively through the left and right atria, meaning the pulmonary vein and the vena cava. They will bring the blood to left and right auricle, from where the blood from the auricle will be filled in now into the ventricle. The semilunar valves are closed at this stage. The SAN now generates an action potential which stimulates both the atria to undergo a simultaneous contraction. Now the atria will contract, because of which the blood flow to the ventricles would increase about 30%, because it is, just imagine it like being squeezed out. Now the action potential is conducted to the ventricular side by the AVN and AV bundle, from where the bundle of His transmits it through the entire ventricular musculature. Clear? This causes the ventricular muscles to contract, because now the ventricular muscles got the signal to do so, and it causes ventricular systole. The atria during this time undergoes relaxation, meaning diastole, atrial diastole. And all of these things happen simultaneously. For example, if the ventricular is if the ventricle is undergoing contraction, then the auricle would undergo relaxation. If the auricles is undergoing contraction, then the ventricles are relaxed. Now, ventricular systole increases the ventricular pressure, causing the closure of tricuspid and bicuspid valves due to attempted backflow of blood into the atria. As the ventricular pressure increases further, the semilunar valves guarding the pulmonary artery on the right side and the aorta on the left side are forced open, allowing the blood in the ventricles to flow through these vessels into the circulatory pathway. Because pulmonary artery and the aorta, they are responsible for transmitting the blood through the whole entire body wherever it is required. So now the ventricles, when they are contracting, the blood will be sent to these blood vessels, and then it will be transferred to the whole entire circulatory, like the whole entire body. Now, after this has been done, the ventricles would now relax, that is ventricular diastole, and the ventricular pressure falls, causing the closure of semilunar valves, which prevents the backflow of blood into the ventricles. Again, the semilunar valves will close down so that no blood can come back. As the ventricular pressure declines further, the tricuspid and bicuspid valves are pushed open by the pressure in the atria exerted by the blood, which is being emptied into them by the vein. The blood now once again moves freely to the ventricles. The ventricles and atria are now again in a relaxed or a joint diastole state as earlier. Again, when the action potential or the impulse will be generated, this whole entire thing will be repeated. This sequential event in the heart, which is cyclically repeated, is known as the cardiac cycle, and it consists of systole and diastole, meaning contraction and relaxation of both the atria and ventricle. As mentioned earlier, the heart beats 72 times per minute. That many cardiac cycles are performed per minute, meaning this whole entire thing of contraction, relaxation, and transferring of blood, this will happen 72 times in 1 minute. From this, it could be deduced that the duration of cardiac cycle is 0.8 seconds when we take this out in seconds. During a cardiac cycle, each ventricle pumps out approximately 70 ml of blood, which is known as the stroke volume. The amount of blood pumped by the ventricle in each cardiac cycle is known as the stroke volume. The stroke volume multiplied by the heart rate, that is 72 beats, gives the cardiac output. So if in the question it comes out to take out the cardiac output, you know how to do so. Therefore, the cardiac output can be defined as the volume of blood pumped out by each ventricle per minute and averages 5,000 milliliters or 5 liters in a healthy individual. The body has the ability to alter the stroke volume as well as the heart rate and thereby the cardiac output. We know that when we exercise, right, when we run, when we jump, our heart rate increases. So this is how our body has this own mechanism by which the cardiac output can be altered. For example, the cardiac output of an athlete will be much higher than that of an ordinary man. Now, during each cardiac cycle, two prominent sounds are produced which can easily be heard through a stethoscope. The first sound is lub, associated with the closure of tricuspid and bicuspid valves, meaning during that time wherein the atrial walls or atricular ventricular walls close down. At that time, the first sound is produced, and that is the lub. Then there is the second heart sound, that is dub. It is associated with the closure of the semilunar walls. Now, when the semilunar walls that are present in between the ventricles and the pulmonary artery and aorta, when they close, then that creates a sound which is known as dub. So, love dub is what dhak dhak is, right? And they are of clinical diagnostic significance because hearing these sounds, the doctor can actually figure it out what is wrong with the heart of the human, if there is or not. Okay. So this is your whole entire cardiac cycle. Now tell me if you have any doubts anywhere. Hmm? Any doubts?
No, ma'am.
The cardiac cycle you've understood?
Yes.
Now moving forward, we have electrocardiographs, ECG. So see, ECG, electrocardiograph and electrocardiogram, both of them are different things. Electrocardiograph is not actually a graph, it is a machine. Okay, now it is a graphical representation of the electrical activity of the heart during a cardiac cycle. What is the electrical activity of the heart? To obtain a standard ECG, this you might have seen in every film which shows the hospital scene. A patient is connected with the machine with three electrical leads, one to each wrist and one to left ankle, that continuously monitors the heart activity. For a detailed evaluation of the heart's function, multiple leads are attached to the chest region. Here, we will talk only about the standard ECG. We want to see the activity of the heart, whether the heart is doing fine or not in terms of the electric activity. So that is ECG. Now, each peak in the ECG is identified with a letter from P to T. P, Q, R, S, and T. That corresponds to a specific electrical activity of the heart. The P wave represents the electrical excitation. Now, this P wave, which is there, it will show that the atria is getting contracted or atricular depolarization, because of which the contraction of atria happens. Now, the QRS complex represents the depolarization of ventricles, meaning this Q, R, and S, it shows that the ventricles are contracting. The contraction starts shortly after Q and marks the beginning of the systole. The T wave represents the return of the ventricles from excited to normal state, which is repolarization. And the end of the T wave marks the end of a systole. So this PQRST is the whole entire cardiac cycle. Now, when we count the number of QRS complexes that occurs in a given time period, one can determine the heartbeat rate of an individual. Since the ECG obtained from different individuals have roughly the same shape for a given lead configuration, any deviation from this graph that we are obtaining, any deviation from it means there is a problem in the heart. So it is of great clinical significance. Just give me 1 second. All right, am I audible?
Yes, ma'am.
Now see, what happens, what doctors do when they look at the screen and read this, they read or they count the QRS, the times that it is appearing, is it not elevated or something, then that tells them that, okay, something is wrong with the patient or not wrong at all. Now we'll talk about double circulation, which happens in all the like chordates and all the higher organisms. Now talking about the double circulation. The blood pumped by the right ventricle enters the pulmonary artery, whereas the left ventricle pumps blood into the aorta. The deoxygenated blood pumped into the pulmonary artery is passed on to the lungs, from where the oxygenated blood is carried by the pulmonary veins into the left atria. This pathway constitutes the pulmonary circulation. See, double circulation has two components. One is pulmonary circulation and the next is systemic circulation. The two types of circulations are there, pulmonary and systemic circulation. The oxygenated blood entering the aorta is carried by a network of arteries, arterioles, and capillaries to the tissues, from where the deoxygenated blood is collected by a systems of venules and veins and vena cava, and they are emptied into the right atrium. This is known as the systemic. See, the blood circulation in between the heart and the lungs is pulmonary circulation, and then the blood circulation in between the body and the heart, that is systemic circulation. The systemic circulation provides nutrients, oxygen, and other essential substances to the tissues and takes CO2 and other harmful substances away for elimination. A unique vascular connection exists between the digestive tract and liver, which is known as hepatic portal system. This is a unique vascular connection in between two organs like a digestive tract and a liver. So we'll call it as hepatic portal system. The hepatic portal vein carries blood from the intestine to the liver before it is delivered to the systemic circulation. So here this is a separate circulation apart from the normal circulation which occurs in our body. And here from the intestine, the blood is brought to the liver and then only it will be delivered to the systemic circulation, meaning to the heart. Now, a special coronary system of blood vessels is present in our body exclusively for the circulation of blood from the cardiac musculature. Now, see, this is the diagram. Starting from here, blood from all the body parts is collected by the vena cava and it is brought to the right auricle. So the right auricle receives the deoxygenated blood. Then from the right auricle, it goes into the right ventricle and then it gets pumped out and it gets carried by the pulmonary artery to the lung. Then from the lungs, after the blood gets oxygenated, it is carried out by the pulmonary vein to the left auricle, then from the left auricle to the left ventricle, and then from the left ventricle, it is carried out by the aorta to the body parts. So this is how the blood is circulated twice in the heart, and this is double circulation. Do you have any doubts?
No.
Okay, apart from this, another thing to remember is that our blood, okay, see, our blood is circulated with the help of the veins and the artery. Now there is a difference between the veins and the artery. What is that difference? The vein has a wider lumen. This lumen part is wide in veins and arteries would have a smaller lumen. Apart from that, all the arteries carry oxygenated blood except pulmonary artery. Pulmonary artery carries deoxygenated blood. And all the veins, they carry deoxygenated blood, meaning CO2 rich blood, except pulmonary vein. Pulmonary vein carries oxygenated blood. Always remember this. It is very important. Now, regulation of cardiac activity. Normal activities of the heart are regulated intrinsically, meaning they are auto-regulated from the inside by the special muscles which are the nodal tissue. Hence, our heart is myogenic. It regulates on its own. A special neural center in the medulla oblongata can moderate the cardiac function through autonomic nervous system, which is ANS. Signals, neural signals through the sympathetic nerves, which is a part of ANSC. Our autonomic nervous system has two components. It has sympathetic and parasympathetic. So the sympathetic nerves, they can increase the rate of heartbeat, the strength of ventricular contraction, and thereby increases the cardiac output. On the other hand, the second part, which is the parasympathetic neural signal, they decrease the heart rate, they decrease the speed of conduction, and thereby the cardiac output. Adrenal medullary hormones, meaning the hormones from the adrenal gland, the medulla part of the adrenal gland, they also increase the cardiac output. So even though our heart can contract, relax, and generate this whole entire thing on its own, but in our body, there are certain mechanisms because of which the cardiac activity can either be increased or decreased. Regulation is clear?
Yes.
Okay. Now we'll come on to the diseases. The disorders of the circulatory system. High blood pressure or hypertension. Now, hypertension is the term for blood pressure that is higher than normal, that is 120 over 80. In this measurement, the 120 mmHg is millimeters of mercury pressure is the systolic pressure or the pumping pressure, and 80 mmHg is the diastolic or the resting pressure. So 120 over 80, this is the normal blood pressure. Now, if repeated checks of blood pressure of an individual is 140 over 90 or higher than that, it shows hypertension. High blood pressure leads to heart diseases and also affects vital organs like brain and kidney. So high blood pressure is when the blood pressure is continuously higher than 140 over 90 mmHg. Now talking about the next disease, that is CAD, that is coronary artery disease. Coronary artery disease, often referred to as atherosclerosis, it affects the vessels that supply blood to the heart. It is caused by deposits of calcium, fat, cholesterol, and fibrous tissue, which makes the lumen of the arteries narrower. See, we know that the lumen of the artery is already narrow. Now, what happens if a person eats a lot of junk food and everything? The deposition starts happening in the artery, because of which the space in the artery is reduced because of this deposition. And this causes the, you can say, the surface area or the lumen to become narrow. Now talking about angina. Now angina is also known as angina pectoris. It is a symptom of acute chest pain when no enough oxygen is reaching the heart muscle. Angina can occur in men and women of any age, but it is more common among the middle-aged and the elderly. It occurs due to the conditions that affect the blood flow. Again, if there is any kind of obstruction in the blood vessel, then that will also cause angina, because when the lumen will be decreased, then not enough oxygen will reach. Now, lastly, there is heart failure. Heart failure means the state of heart when it is not pumping blood effectively enough to meet the needs of the body. It is sometimes called as congestive heart failure because the congestion of lungs is one of the main symptoms of this disease. Heart failure is not the same as cardiac arrest. Cardiac arrest is when heart stops beating, or a heart attack. Heart attack is when the heart muscle is suddenly damaged by inadequate blood supply. So heart failure, or we can also call it as congestive heart failure, then cardiac arrest, and then there is heart attack. Three of them look the same, sounds the same, but all the three of them are different. So with this, we have completed the chapter. Have you understood the disorders, how they happen?
Yes, ma'am.
Any doubt?
Yes, ma'am. Ma'am,
Hmm.
can you once again explain what is the hepatal portal system?
Hmm.
No, what do you mean by the portal system in general?
See, port. What is a port? Port, for example, airport. What is it? A platform wherein different airplanes come and stay, isn't it? It sort of connects the different locations at one point, isn't it? So same way the portal system connects organs together. The hepatic portal system is a portal system which will connect two organs together. One is the digestive tract, the whole entire digestive system, with the what? To the liver. See, anything related with the liver is known as hepato or hepat, because the liver is made up of hepatocytes. Okay, so that is why anything related to the liver will be hepato, anything related to the heart will be cardio, then anything related with the lungs will be pulmonary. Okay, so hepato is a word that is denoting that it is a portal system which is connecting it to the liver. So that is why hepatic portal system. Here what happens, the digestive tract and the liver, they are sort of connected, and this portal vein, the vein from this hepatic portal system, it carries blood from the intestine, which is again a part of the digestive tract, and it carries from the intestine to the liver before it delivers it to the systemic circulation. This is sort of like a junction wherein first the blood is collected from the small intestine, and then it goes to the liver, and from the liver it is delivered to the heart. It does not go direct. Clear?
Yes, ma'am.
Any doubts?
No.
Hmm. Okay, now you do one thing. Since the chapter is over, take 5 minutes, read the book, and tell me if there is anything that is troubling you. Else we'll move on to the new chapter. Yes, any doubts?
No.
Okay. So now let's start a new chapter. Is my screen visible?
Yes, ma'am.
Okay. All right. So now let's start with a new chapter, that is this thing, excretory products and their elimination. Now, animals accumulate ammonia, urea, uric acid, carbon dioxide, water, and ions like sodium, potassium, chlorine, phosphate, sulfate, etc., either by metabolic activities or by other means like excess ingestion. We start accumulating these stuff in our body. Naturally, it happens. Now they have to be removed from our body because their accumulation for a longer period of time will cause a lot of problems in our body. So for that very reason, it has to be actually eliminated in our body. So in this chapter, we'll talk about how these things are eliminated, and also we'll talk about the very common nitrogenous bases. Now, ammonia, urea, and uric acid are the most common types of nitrogenous base excreted by the animal. Ammonia is the most toxic form and requires large amount of water for its elimination, whereas uric acid is the least toxic and can be removed with a minimum loss of water. This is important to remember, that which nitrogenous waste is the most toxic and which one is the least toxic. Now, the process of excreting ammonia is ammonotelism, and those organisms that excrete ammonia, they are ammoniotelic, such as bony fishes, aquatic amphibians, and aquatic insects. All of them are ammoniotelic in nature. Ammonia, as it is readily soluble, it is generally excreted out by diffusion across the body surfaces or through the gill surfaces in a fish as ammonium ions. Kidneys do not play any significant role in its removal. Now, terrestrial adaptation necessitated the production of lesser toxic nitrogenous waste such as urea and uric acid, so that water can be conserved in them, because for terrestrial organisms, they don't have a long source of water for them. So what they do as a mechanism, they excrete out less toxic nitrogenous waste. Now the mammals, many terrestrial amphibians and marine fishes, they mainly excrete urea, and they are known as ureotelic animals. Ammonia produced by metabolism is converted into urea in the liver of these animals and then released into the blood, which is filtered and excreted out by the kidney. The blood filtering happens in the kidney. Now, some amount of urea may be retained in the kidney so that to maintain the osmotic balance. Now talking about the reptile, the birds and the land snail and the insects, they excrete the nitrogenous waste in the form of pellet or paste with the minimum loss of water, and they are uricotelic because they excrete out uric acid. So we have ammoniotelic, ureotelic, and then uricotelic, three types of animals. A survey of animal kingdom presents a variety of excretory structures. In most of the invertebrates, these structures are simple tubular forms, whereas vertebrates have complex tubular organs known as kidneys. Some of these structures are mentioned here. Protonephridia or flame cells are excretory structures in platyhelminths. Protonephridia or flame cells. Platyhelminthes are flatworms such as planaria. Then rotifers and some annelids and cephalochordates such as Amphioxus and Amphioxus, they also have the flame cells or protonephridia. Now, the protonephridia are primarily concerned with ionic and fluid volume regulation, that is, osmoregulation. Nephridia are tubular excretory structures of earthworms and other annelids. Nephridia help to remove nitrogenous waste and maintain a fluid and ionic balance. Malphizian tubules are the excretory structures of most of the insects, including the cockroaches. Malphizian tubules help in the removal of nitrogenous waste and osmoregulation. Then there is antennal glands or green glands perform the excretory function in the crustaceans like prawn, which is a member of the class Insecta. So these are the variety of excretory structures, and the best thing to note about them is that apart from excretion process, they also excrete they also do the process of osmoregulation. Okay, any doubts up until now?
No, ma'am.
All right. Now moving forward, let's talk about the human excretory system. Now, human excretory system consists of a pair of kidneys, one pair of ureters, and a urinary bladder, and then there is the urethra. Kidneys are reddish brown, bean-shaped structures situated between the levels of last thoracic and third lumbar vertebra. Now, see, in our body, there is like the presence of vertebrae. Now, in the last thoracic and the third lumbar, there is where the kidneys are situated. Now, they are situated close to the dorsal inner wall of the abdominal cavity. Each kidney of an adult human measures about 10 to 12 centimeter in length, 5 to 7 centimeter in width, and 2 to 3 centimeters in thickness, with an average weight of 120 to 170 grams. Towards the center of the inner concave surface of the kidney is a notch which is known as hilum, through which ureters, the blood vessels, and the nerves would enter. Inner to the hilum is a broad funnel-shaped space that is known as renal pelvis, with projections that are known as calyces. So now we are talking about inside the kidney. Now, outer layer of kidney is a tough capsule. Inside the kidney, there are two zones, the outer cortex and the inner medulla. The medulla is divided into a few conical masses, cone-like structures that are known as the medullary pyramid, that project into the calyces. The cortex extends in between the medullary pyramids as the renal columns, called as the columns of vertebrae. See this, the cortex extending here. Right in between will become the renal columns or the columns of vertebrae. Just 1 second. Now, each kidney has nearly 1 million complex tubular structures known as nephrons. Now, nephrons are the functional unit of the kidneys. Each nephron has two parts, the glomerulus and the renal tubule. The glomerulus is a tuft of capillaries formed by the afferent arteriole. Afferent is the one that is coming. Okay, and then which is the renal artery, and the blood from the glomerulus is carried away. It goes out of the glomerulus by the efferent arteriole. So there are two types of arterioles, afferent and efferent. Now, the renal tubule begins with a double-walled cup-like structure called as the Bowman's capsule. Now, that glomerulus that we talked about is present inside the Bowman's capsule only. And the Bowman's capsule, along with the glomerulus, is known as the Malpighian body or the renal corpuscle. This is the name. Now, the tubule continues further to form a highly coiled network that is known as PCT, the proximal convoluted tubule. Meaning extending from here, we have the proximal convoluted tubule. A hairpin shaped Henle's loop is the next part of the tubule, which has a descending and an ascending limb. Now, the PCT or the proximal convoluted tubule will continue with the descending loop of Henle and then to the ascending loop of Henle. Descending is downwards, ascending is upwards. The ascending limb continues as another highly coiled tubular region, that is DCT, distal convoluted tubule. The DCTs of many nephrons opens into a straight tube called as the collecting duct. Then several DCTs open into a collecting duct, many of which converge and open into the renal pelvis through medullary pyramids into the calyces. The Malpighian corpuscle, the PCT and the DCT, all of them are situated in the cortex of the kidney. So they are cortical, whereas the loop of Henle goes into the medulla. In majority of the nephrons, the loop of Henle is short and extends very little into the medulla. So those type of nephrons are known as cortical nephrons, which do not dip deep inside the medulla. They are cortical because they remain up till the cortex only. Now, in some of the nephrons, the loop of Henle is very long and runs deep into the medulla. These nephrons are called as juxtamedullary nephrons, the ones that are dipping deep inside into the medulla. Clear? Up until now, do you have any doubts?
No, ma'am. Yes.
Everything is clear?
Yes.
Is it getting hodgepodge?
No.
No? Sure? Shall I continue?
Yes.
Okay, very good. Now, the efferent arteriole, which emerges from the glomerulus, efferent, the away one, it forms a fine capillary network around the renal tubule, which is known as the peritubular capillary. Now, a minute vessel of this network runs parallel to the Henle's loop, forming a U-shaped vasa recta. Vasa recta is absent or highly reduced in cortical nephrons. So, why is it highly reduced in the cortical nephrons? Because it does not dip into the medulla part. And since it does not dip into the medulla part, so that goes and coincides with the cortex or the cortical nephrons, because they are not dipping into the medulla. They stay in the cortex region of the kidney. Now, let's talk about the urine formation. It has 3 steps: the glomerular filtration, reabsorption, and secretion. It takes place in the different parts of the nephron. The first step in urine formation is the filtration of blood, which is carried out by the glomerulus, and it is known as glomerular filtration, the first step. On an average, 1,100 to 1,200 ml of blood is filtered by the kidneys per minute, which constitutes roughly 1/5 of the blood pumped out by each ventricle of the heart. So the glomerular capillary blood pressure causes filtration of blood through 3 layers. See, the filtration of blood, it is a unit, okay, which is made up of 3 layers. The endothelium of the glomerular blood vessel, the epithelium of the Bowman's capsule, and a basement membrane between these 2 layers. The epithelial cells of Bowman capsule, they are known as podocytes. They are pore-like structures and they are arranged in an intricate manner so as to leave minute spaces called as filtration slits or slit holes. Blood is filtered so finely through these membranes that almost all the constituents of the plasma, except the proteins, because proteins are large size molecules, they pass on to the lumen of the Bowman's capsule. Therefore, it is considered as ultrafiltration. Imagine it being a greatest filter paper-like structure because of which the filtration of all the components of plasma happens except for the protein, because proteins are obviously very large size molecules. Now, the amount of filtrate formed by the kidneys per minute, meaning the amount of plasma or the amount of blood that has been filtered by the kidney per minute, is known as glomerular filtration rate, GFR. GFR in a healthy individual is approximately 125 milliliters per minute. That is 180 liters per day. Now, kidneys have built-in mechanism for the regulation of GFR. One such sufficient mechanism is carried out by juxtaglomerular apparatus. An apparatus is there that carries out the filtration of the kidney, the ultrafiltration, and then how the GFR is maintained in the body. Now, juxtaglomerular apparatus, it is a special sensitive region formed by cellular modification in the DCT and the afferent arteriole, meaning the point where the DCT and the afferent arteriole contacts each other. At that point of time, there is the presence of juxtaglomerular apparatus. A fall in the GFR. As soon as the glomerular filtration rate decreases, meaning it becomes less than 125 milliliters per minute, then what happens? The cells of the juxtaglomerular apparatus, they get activated to release renin, which causes the glomerular blood flow to get stimulated, and it will bring the GFR back to normal. Now, comparison of the volume of the filtrate formed per day. Per day, 180 liters of filtrate is formed with that of the urine release, that is 1.5 liters. Suggests that nearly 99% of the filtrate has to be reabsorbed. This process is known as reabsorption, the second process of urine formation. Like even though the filtrate formed per day is 180 liters, but still hardly 1 liter of urine is passed out. So it means that 99% of the filtrate gets reabsorbed. The tubular epithelial cells in different segments of nephron perform this either by active or passive mechanisms. For example, substances like glucose, amino acids, sodium in the filtrate are reabsorbed actively, whereas the nitrogenous wastes are absorbed by the passive transport. The reabsorption of water also occurs passively in the initial segments of the nephron. During urine formation, the tubular cells secrete substances like hydrogen ions, potassium ion, and ammonia into the filtrate. Tubular secretion is also an important step in the urine formation as it helps in maintaining the ionic and acid base balance. So, talking about the three basic steps, ultrafiltration, then there is reabsorption, and lastly there is secretion. So, this is your the way by which the urine formation takes place in us humans. Any doubts?
No.
Okay. Now let us talk about the functions of the tubules that we had discussed. So the function of PCT, proximal convoluted tubule. PCT is lined by simple cuboidal brush border epithelium. Type of epithelium which is present in the PCT is simple cube-like epithelium. This increases the surface area for reabsorption. Nearly all of the essential nutrients and 70 to 80% of electrolytes All of the essential nutrients and 70 to 80% of the electrolytes and water are reabsorbed by this segment. PCT also helps to maintain the pH and ionic balance of the body fluids by selective secretion of hydrogen ions. Just give me 1 second. All right. Harshad, I think your class starts half an hour later than this. Okay.
Ma'am, at 6,
Yeah. What time is it at your place right now?
5:30.
5:30. Yeah. So it says in my schedule that your class starts half an hour later. Just let me confirm and then I'll tell you right now. Okay. Just let me confirm.
1 second, ma'am.
Yeah. You also check whether you have joined wrong because in my schedule your class starts half an hour later than this.
Yes, ma'am, the class is at 6.
Yeah, so you join half an hour later from now, okay?
All right, ma'am.
Okay. All right, so yeah, I was discussing about the PCT, that is the proximal convoluted tubule. Now, this is lined by the simple cuboidal brush border epithelium, which increases the surface area for reabsorption. Nearly all of the essential nutrients and 70 to 80% of electrolytes and water are reabsorbed by this segment. PC